Hi, I'm Sietse and I'm standing in front of the Agrico Research Center. Agrico is one of the biggest Dutch potato companies, with locations in more than 80 countries around the world. Agrico played a big role in helping us develop our iGEM project. Let's head inside. According to the Food and Agriculture Organization of the United Nations, over $750 billion worth of food is lost every year before it can be consumed. To put that into perspective, that is enough to solve world hunger 3 times over. These numbers are largely due to pests and pathogens, nematodes being one of them. We are Team iGEM Groningen and we are here to put a stop to it. The potato is one of the biggest contributors to the Dutch agricultural sector. It occupies 1/4 of the country's farmlands and receives half of all Dutch agricultural subsidies. In 2017, the Netherlands exported $100 billion worth of agricultural goods, and potatoes contributed $1 billion to this number. This clearly shows how important this crop is for the Netherlands. Parasitic nematodes are the biggest threat to this industry. Among these nematodes, the biggest troublemaker for Dutch farmers is Globodera pallida. G. pallida is native to the Andes and is now present in 55 countries, predominantly in temperate regions. In Europe alone, this nematode causes crop losses of $460 million a year.
Hi, my name is Fenny, and now I will walk you through the life cycle of Globodera pallida. It involves multiple stages and begins with the eggs hatching in the presence of root secretions called exudates. When the eggs hatch, the nematodes enter the juvenile stage and move towards the root exudates. The nematodes penetrate the root tissue of the potato plant and form feeding sites inside the root that are necessary for their survival. When they enter the adult stage, the nematodes consume the nutrients and the water that the potato needs to grow and develop. The females, after fertilization, develop into new cysts, giving rise to 200 to 500 new nematode eggs. In this form, they can survive in the soil for up to 20 years, waiting for the favorable conditions and presence of a potato plant as a new host. There are several methods being used nowadays to fight off the G. pallida in the potato field. However, no method so far has been proven to be fully effective and sustainable. The use of pesticides is a common practice, but it's often detrimental to soil biodiversity. It is therefore slowly being phased out by governmental institutions around the world. Sometimes, farmers apply drastic measures, like flooding the whole field. Although it is effective, this method is very costly for the farmer and is not only limited to G. pallida. Crop rotation, the shifting of the crop grown every year by the farmer, is popular but not fully protective, and not every farmer is capable of doing this. Classical breeding methods can be an effective method, but it takes too long to develop because of the adapting G. pallida population. Genetically modified crops are developed faster, but both consumers and governmental institutions don't really consider this to be a safe solution. Considering all of these issues, we decided to develop a different solution, RootPatch. While developing RootPatch, we needed it to hold 4 key values. Firstly, since farmers need to apply it on a large scale, RootPatch should be easily applicable. Second, we aimed at developing a GM solution, so we had to ensure its safety and control to avoid any spread of GMOs in the environment. Third, RootPatch should only affect the harmful nematode, keeping the beneficial organisms in the ecosystem intact. And last but not least, since G. pallida is not the only parasitic nematode in the world, we aim to develop a general solution that could be applied to other parasitic nematodes in the soil. We were brainstorming about how to solve the nematode problem when we got an idea. What caught our attention was the work of Jonathan Dalzell and his colleagues, who explored the effects of neuropeptide-like proteins on the chemotactic behavior of nematodes. Neuropeptide-like proteins, or simply NLPs, can be found in many species of nematodes as they are involved in the regulation of their neuronal behavior. NLPs are different from the classical neurotransmitters in humans because they can modulate the activity of a whole neuronal network, rather than just facilitating single neuron-to-neuron communication. This is also why they are believed to be very significant for the nematode's functioning. NLPs also regulate the neuronal networks for sensing the environment, causing the nematodes to be attracted by the root exudate. The group of Dalzell attempted to investigate how increasing the levels of the NLP in G. pallida would affect their chemoattraction towards the exudates. They investigated this with a simple experiment. After putting the nematodes in contact with different NLPs for 24 hours, thereby changing their internal NLP levels, they were placed in the middle of a petri dish. This petri dish was divided into 3 parts, one with water, one with the root exudate, and a neutral stroke in the middle. By monitoring the movements of the nematodes on this petri dish, they were able to point out NLPs that could be used to reverse the attraction of the nematodes towards the root exudates. They identified NLP14A to be efficient in reversing the chemoattraction, and importantly, also showed that this NLP appears to be specific in only parasitic nematodes. The next problem we faced was a method to deliver the NLP into the soil, where it would meet the nematodes. In order for the neuropeptides to always protect the roots, they need to be constantly present in the rhizosphere. Chemical synthesis of NLPs and its application wouldn't suffice, as this would require a constant application and so many man-hours. Therefore, we reasoned that the NLPs should be produced at the roots directly. Next, we need to select the host organism for the NLP production. We were divided between engineering the potato plant or a soy bacterium. We didn't want the potato itself to be engineered due to the consumer's reluctance to eat GM crops. So by choosing a soil bacterium, this gave us 2 benefits. First of all, soil bacteria tend to form biofilms around the roots, providing structural and nutritional support for both the bacteria and the plants. Secondly, the same solution can be applied to other plants without having to engineer anything new.
After we decided that we were going to use a bacterium as the RootPatch host, our first choice was Bacillus subtilis, since it's a well-studied soil organism. However, because we were going to genetically modify it, we did not know how robust it would be in the soil. That is why we reached out to academic experts. The first person we contacted was Professor Oskar Kuipers from the University of Groningen. He strongly suggested that we use a dominant soil organism because the applications would be very limited if it would get outcompeted. He directed us towards the work of one of his students, Yang Lai Yi, who studied the effects of potato root exudate on Bacillus mycoides. He also suggested that we talk to Jan Spoelder, an expert in phytopathology and the application of biological agents. Based on his advice, we used a Bacillus mycoides strain called M2E15, which is dominant, spore-forming, and also naturally forms biofilms on the roots of potatoes. So when we had decided on a host organism, we then had to think about how to introduce RootPatch into the soil. Based on the advice from Jan Spoelder, we decided on a solid bioformulation, since these are easier to apply, easier to store, and cheaper to produce than liquid bioformulations. After speaking to the farmers, we realized that the equipment needed to apply a powdered bioformulant already exists, which is very advantageous for us. The RootPatch granulate would have to be applied during the potato planting in April. This would give the RootPatch a chance to establish itself on the tuber and then grow along the forming potato roots.
Sietse shared our plans for the formulation and application of RootPatch with you. Now let's get back to the synthetic biology part. To study whether RootPatch would actually work, we needed to perform experiments. But because of the corona pandemic, we couldn't actually do this. So we had to look for alternative ways to study how RootPatch would perform. We shifted to a computational approach by creating models that can incorporate how the environment could affect how well RootPatch would perform. Humidity, temperature, and pH all have an impact. And by using computational power, we can study and expose any possible weaknesses. A model studying the bacteria population in RootPatch showed the importance of several factors. The initial phase of growing RootPatch showed to be the most sensitive. Ensuring that RootPatch would survive here allowed it to survive throughout the whole season. Also, competition has a great impact on the survival of RootPatch throughout the season, and is therefore important to ensure that our host bacterium is capable of surviving the competition in the soil. By choosing a natural soil isolate, we are hoping to achieve this. Overall, the most important environmental factor turned out to be humidity. The lower the humidity, the harder it is for RootPatch to survive. But all in all, our results show that RootPatch is strong enough to survive in the soil throughout the whole potato season. Safety is an important consideration when using free-living GM bacteria. While the model showed that RootPatch is strong enough to survive, we only want its presence to be at the roots. To ensure this, we developed two safety mechanisms that are dependent on the presence of specific components in the soil. Tryptophan and solanine specifically are way more abundant in the potato plant exudates than in the exudate of any other plant. By making RootPatch dependent on them, we can allow it to survive only in this environment. To make RootPatch dependent on tryptophan, we decide to make it oxotrophic for this amino acid by knocking out the TRPE gene, which is required for the metabolism of tryptophan. Because other plants produce tryptophan as well, this will not be entirely effective, but it will maintain the bacteria already to a certain extent. To improve the containment, we will make the bacteria dependent on solanine as well, which is specific to the potato plant. A toxin is introduced into the genome of the bacteria, and its antitoxin will only be expressed when solanine is in the environment. Hopefully, the combination of the two strategies will be enough to keep RootPatch where it belongs. Now let's take a look at RootPatch and how it is engineered to produce the neuropeptides. We plan to introduce the NLP14A gene into the chromosome of B. mycoides using a CRISPR-Cas9 technique. A PYCR vector, which contains the NLP14A gene and the Cas9 protein required to cut the genome open, is introduced into the bacteria. The vector will integrate the gene into the genome behind a strong constitutive PTA promoter. This promoter will ensure the expression of the gene and will allow a constant neuropeptide presence around the roots. Considering RootPatch can now express the NLPs, let's see how they act on the nematodes. The nematodes hatch and start moving towards the roots after sensing the root exudate that has been released into the soil. But once they are close to RootPatch, the NLP will enter the nervous system of the nematodes, changing their neuronal activity and now causing them to avoid the exudates of the root. To understand how the application of RootPatch affects the population dynamics of G. pallida throughout the season, we developed a nematode population model. Several key factors play an important role in the ability of RootPatch to protect the root from the nematode invasion. With a higher number of nematodes in the soil, higher NLP production is required to protect the plant. High soil temperatures speed up the life cycle of the nematodes, making them move faster through the soil, leaving less time for RootPatch to react. But most importantly, the efficacy of RootPatch will depend on the ability of the nematodes to go back to their normal functioning after leaving the NLP environment. Future experimental data can help this model really predict how effective RootPatch actually is. As I mentioned earlier, the corona pandemic denied us access into the labs, and since we are unable to produce experimental evidence, we could not confirm some of the key aspects of our project. Questions like whether the bacteria will survive in the soil, or whether NLP14A will be effective enough to repel G. pallida are still left unanswered. Because of this, we made a decision to participate in the first ever two-phase iGEM project. In order to give the iGEM 2021 Groningen team a head start for the next year, we have set up complete wet lab plans. We have also made models to expose weaknesses and strengths of RootPatch. We've established collaborations with national laboratories, and we learned critical information about the problem by talking to various stakeholders and setting up communication with a number of potential future sponsors. to realize that the farmers that we spoke to were open to using RootPatch in their fields as long as it is allowed by their governments. When we spoke to Sjaak Aben from Lam Weston, which is a potato processing company, we were assured that RootPatch can definitely be a solution for the current G. pallida problem. However, there will be hurdles like legislation and public acceptance that need to be overcome. For our outreach program, we contacted Niels Alberts, who works for Science Links, an educational institution that promotes science to the public. He introduced us to Nemo, the biggest science museum in the whole of the Netherlands, which also co-hosts the DNA Dialogue. The DNA Dialogue is a national collaboration of influential Dutch institutions, like the Dutch Institute for Health and Environment and the Erasmus Medical Center. The aim of this initiative is to discuss the ethical implications of human genome editing in embryos. Due to the pandemic, all of their personal events were canceled, and the DNA Dialogue had to be moved online. Our iGEM team designed an online educational platform to still make the DNA Dialogue possible, even in times of social distancing. On this educational platform, we host 2 lessons, accessible for everyone who is interested. Our first lesson provides the learner with a fundamental background on CRISPR-Cas genome editing and its possible applications to treat genetic diseases. In the second lesson, the ethical dilemmas of editing human embryonic DNA will be discussed. The overarching aim of this lesson series is to let the learner form an educated opinion on the use of CRISPR-Cas on human embryos. The unique features of the lesson series are that it is in Dutch and English, so that it can be applicable to a wider audience. It also includes a questionnaire of the Erasmus Medical Center, which collects opinions of the course participants on genetic modification of human embryos. The results of the questionnaire will be used by the Dutch government to create policies on the use of genome editing techniques. By putting our lessons online, we make sure that the activities of the DNA Dialogue continue even during the pandemic. However, our outreach activities this year were not limited to developing this lesson series. We also helped in Discovery Day, where Dutch kids would explore science and perform cool experiments together with supervisors. Our project was featured in EU SynBios and biotechnology.nl blogs, several journals, and interviews, giving us plenty of opportunities to make the people familiar with RootPatch. Now we go back to Siets in the field, where he talks about the future goals of RootPatch.
So the fate of RootPatch is still uncertain. Its future depends on the rules and regulations of the EU and how it is accepted by the public. We paid close attention to these concerns during our project, and we talked to farmers, industry experts, and the public. We realized that RootPatch is so much more than just potatoes and G. pallida. If proven safe enough, RootPatch could show the way to new methods of protecting plants using genetically engineered bacteria. In addition, the neuropeptides could provide a more sustainable method of crop protection. But before we get there, we need to dive into the lab and show that this is actually true. We hope to inspire future iGEM teams to explore similar approaches to RootPatch to help combat drastic crop losses worldwide. In the end, we would like to thank everyone who supported us during this amazing project.